Thank you for your introduction. I'm Ruyi Chen, a PhD student at the University of Queensland. Today, I'm going to present one of my PhD projects, learning the languages of phylogeny with MSA transformer, and you can scan the code to access our preprint here. So I think a long-standing question in biology is to learn the evolutionary relationship from the sequence data. And in the previous half centuries, people rely on the multi-sequence alignment to build so-called never join or the maximum likelihood trees. However, the big issue of this method is the assumptions. It generally assumes that each alignment site evolved independently and didn't consider the precedence of the impedance. So to address these issues, we want to use the protein languages models. When training the model, we would usually randomly mask 15% of the tokens. And in this way, our model was asked to recover or to reconstruct the probability of the mask tokens. So people will learn the dependence of different columns. And learn the epistics. Based on these assumptions, and we want to use a more specific model called the MSA transformer. It takes a multi-sequence alignment as input, and is first maybe published in the 2021 ICLR conference, and it is consists of the 12 layers. And for each layer, it also have row attention, column attention, and the embeddings. So the previous research indicates that the row attention encode some structural contact, while the paper that published in Nature Con show that the column encode some phylogenetic relationship. So based on the previous research, we want to dig more about how this row attention or the embeddings to capture some phylogenetic relationship. So our question is, if it's capture the phylogenetic relationship, the MSA transformer rely on what kind of information to capture it? And the second question is, if we can use the MSA transformer to build the tree, are the tree generated by the MSA transformer comparable to the traditional phylogenetic trees? So with these questions, we do our experiment design. We collect the original multi-sequence alignment from the PFAM database. And apart from that, we also generate 3 scaffold multi-sequence alignment that either preserve or break the conservation information within the columns. So now it has 4 different multi-sequence alignment. The previous 2 preserved the conservation information, and the last 2 break the conservation information within the columns. And then we extract the embeddings and the column attention head using the MSA transformer layer by layer, and then use this original multi-sequence alignment. We generate the never join and the maximum likelihood tree. And so this is a more detailed step about our experiment design. For each multi-sequence alignment tab, we first compute the sparse membrane correlation of the 2 components, that is column attention and the embeddings with the evolutionary distance calculate from the never join or the maximum likelihood tree we have already built. And then we use the workflow here. We have calculated this distance matrix and perform the never join algorithm to build the so-called embedding tree and the attention tree. And the next step is we try to benchmark the similarity between the MSA transformer trees and the classical phylogenetic trees by using of the clustering information score and the average across the 20 protein families. The clustering information score measures some similarity with our inferred trees and the traditional phylogenetic tree. The higher score means the higher similarity. So this is result about in the column attention space, and we could find the column attention heads are correlated with the evolutionary distance, and most column attention heads are negatively correlated with the evolutionary distance. However, we could find some attention heads, such as the head from layer 1 and the head 5, or the layer 12 had 10, it will be positively correlated with the evolutionary distance across 20 protein families. And then we find if this colon attention head is positively correlated with evolutionary distance, we use this attention head to build the tree. The tree is similar to the never join and the maximum lived tree. So this is what this heat map tell us. And so this is the result about the colon attention space. And for our embedding space, we find the distance between the pair of the sequences in the embeddings all positively correlated with their evolutionary distance. And the second is we find that if the embedding trees generated from the early layers would have the more similarity to the never join and the maximum lived trees. Apart from that, we also find for both attention trees and the embedding trees, if we build the tree using the previous two multi-sequence alignment, it have the comparable result and very similar to the traditional phylogenetic trees. And if we build the tree using the last two multi-sequence alignment, it have the distinct patterns. So it may suggest that it rely on some, maybe the information within the columns, or we call the conservation information to learn the evolutionary relationship. So to further explore this, here we directly compare the similarity of the embedding trees, the similarity of our embedding trees generated by the original multi-sequence alignment and the other three shuffled multi-sequence alignment. And here is our result. So we first find the similarity between the embedding tree generated from the original multi-sequence alignment and the shuffle column multi-sequence alignment successively decrease across the layer. So in this step, we just compare this tree with this tree. And the second step, secondly, we find the embedding tree generated from the other two multi-sequence alignment shows significant lower similarity to those from the original multi-sequence alignment. So the huge differences between these plots further indicate that the MSA transformer primarily rely on the conservation information or learn the tree topology or the phylogenetic relationship. So you may know this way is actually very like we do we construct the trees, do the phylogenetic research, because our assumption is each column evolve independently. So we focus on the conservation information within the columns. And so when we got this, next step, we thinking about, could we use the embedding tree alone to do some phylogenetic research? So here we show one of the case studies. We first test the accuracy of the embedding trees to do the phylogenetic research. We first consider the RDRD domain. It's only conserved domain across all the RNA virus, so it's used for the phylogenetic inference. This is ML trees that we directly collect from these papers. And then we used based on the same multi-sequence alignment and used our method. We built the corresponding embedding trees. And here I just presented the embedding tree built from at the early layers because it's more similar to the traditional phylogenetic trees. And we find the high concordance between the embedding tree and the ML trees. And our embedding tree have a very clear groupings of the virus. So we may think the embedding tree may be robust and it can be further used for some phylogenetic research. And we find the quite similarity between the two trees. The next question is, what's their differences? If they are all similar, what is advantages or the disadvantages of the MSA transformer trees? So we are not satisfied with this. Further, we want to use the embedding tree to address more complicated evolutionary problems. So here we consider the large DNA virus. One of the key challenges in these evolutions is to determine whether they're conserved proteins is to determine whether their conserved proteins have the shared evolutionary histories or if some were acquired rapidly from their host. So in the previous research, the authors just identified the eight conserved proteins and built the ML trees. So this is all the ML trees they built. And then, but their expectation is this 8 protein ML trees could demonstrate some monophyletic relationships, but they didn't got this result. Based on the same multi-sequence alignment, we built our embedding trees. We also display the embedding trees from the early layers, and we find our embedding trees has some different result. For example, we compare the embedding trees generated from the TF from the ML tree, they are not grouped together. But when we recover this tree using our method, they really the same colors means the same virus of the same DNA virus grouped together, and this is meet the author's expectations. But it also have some differences. Is if we use the multi-sequence alignment contain a lot of gaps, our embedding virus with another members from other viral species. So the big differences between the embedding trees or the traditional phylogenetic tree is the embedding tree is more sensitive to the gaps, and in the traditional method they usually ignore. So here we got some summaries. Is we think the embedding tree generated from the MSA transformer can capture some dependence between the columns, and then it can also model gaps, although it's not, we don't know if it's correct or incorrect to model gaps, or modeling gaps if it will improve the phylogenetic research. So hopefully we want the embedding trees and the traditional method will complement each other to display the evolution of the proteins. Finally, I want to thank my supervisor, Michael Bowden, and all the members and Bowden Lab at the University of Queensland. I'm very happy to take any questions. Thank you.
Well, that was a very interesting talk. Are there any questions?
Thank you. Very interesting talk. I had a question. When you compare your embedding trees with the ML trees, so you showed there was a strong similarity between both. I was wondering about the differences. Do you see the differences rather distributed uniformly in the trees, or rather at the root compared to toward the tips of the trees?
Do you mean this plot?
Yeah, the location of the differences.
So I think the differences between the traditional method didn't model the dependence between the columns, and the second is the embedding trees will use the gap character when they train the model. So usually they will treat the gap character as a spatial character rather than ignore it. So this is why we use the two different methods, but got similar result, also got some different result. So I think the main differences would be the MSA transformer model epistic effect, and the second is the MSA transformer takes the gap character into consideration.
Thank you.
Yeah, here.
Sorry.
Thank you. I've got a question for yes, just the slide. You said the gap, the transformer. The transformer will be sensitive to the gap. So how long this gap for the base or the amino acids?
How long?
How
I
many?
know.
Yeah,
so
how many?
So in our update manuscript, we find for these sequences, it have the long gaps, and for the other for the sequences from other virus, they have the complete multi-sequence alignment, and only for these sequences, the length of this is half of the total multi-sequence alignment length. So when we cut this multi-sequence alignment to the half length, and then we regenerate the tree, we find the evolutionary information here is the same as the maximum likelihood method. So, but we didn't update our the newest result. But I can tell you that is, if we cut this multi-sequence alignment length to the half length as the original one, it will have the same result, the same phylogenetic evolutionary relationship. Yeah.
Sorry, and I mean the gaps, the range of the gaps.
Yes.
Yeah. How many bases or amino acids?
20 amino acid.
Okay, thank you.
Thank you very much for this talk. Does your embedding can identify correlated mutations in your alignment in the tree?
Sorry?
If you can identify it from the alignment correlated mutations, as you are capable to understand this launch.
Sorry. I think so. So we calculated the evolutionary distance because the previous papers used the Hamming distance. We know Hamming distance is editing distance and is not accurate to portray the evolutionary relationship. So we, based on the previous research, we find we used we find the evolutionary distance will be more accurate to measure some the evolutionary relationship. So we collect. So for our experiment design, we collect the neighbor join and the maximum likelihood tree, and then we calculate the pairwise evolutionary distance, and then we also use the workflow here to calculate their pairwise Euclidean distance of the embeddings. So they will have the same shape, and then we use the Spelsman correlations to measure their correlations. And yeah, so this is the result.
We have time for one more.
I'm trying. Thank you very much. I'm trying to understand. This is working alignment by alignment. So you have one alignment for one protein in the case of the virus, for instance, and then you have another alignment, and every single alignment is going to the transformer. Is that right?
Yes, so I think the specialty of this model is it takes a multi-sequence alignment as input, and also the multi-sequence alignment is what we rely on to build the tree. So we find they all based on the multi-sequence alignment, and if we build the tree using the MSA transformer, can be further transformed to the tree or similar to the tree that we built like the maximum likelihood tree. So we just find the two approach all takes the multi-sequence alignments input. So there should be some similarity or differences. So this is why we do this research.
Is there a way to compare this gene by gene transformer thing to one another and to a kind of a collection? Like if you were to put all the genes together and kind of have a species phylogeny, and do the distance to the species phylogeny will tell you something about how good the transformer, gene by gene transformer.
So quite a good question. So because this is a protein languages models, and we hope the future work would be developing the relevant or equivalent model that will work on the genome in the genomes space. And I think also we should try to design some algorithm to model some interaction between gene and gene or protein and proteins. But this model is not designed for phylogenetic reconstruction. Maybe we should, if the future work, we should design a more complicated model that will meet your requirement.
Okay, well, thank you very much. That was a very